Good morning, everyone. This is the last presentation in session 2, and I'm going to talk about the approaches we have been working on to evaluate the impact of formulation differences on performance of topical products. The objectives for my presentation are first to describe consideration for formulation development of a test product that does not meet the no significant difference criterion. Second, to evaluate the impact of compositional differences or Q2 differences on bioavailability of topical gels. And the last is to evaluate the impact of Q2 changes on sensory perception of topical gels. So you heard from Dr. Kilchan's presentation earlier in this session about bioequivalence approaches that are currently recommended in our published PSGs for topical products. In this diagram, you can see the potential strategies for BE that was developed or are under development under GODUFA Regulatory Science Program. And they have grouped them based on the requirement in the for the formulation. So when a test product, for example, meets the no significant difference criteria and it is pre-qualified for a characterization-based approach, and this approach is currently recommended in many of our PSGs for topical products. But when there are differences in a formulation of a test product compared to reference standard, then there are other approaches that we have been working on to develop for this category of test formulation. So that there are approaches based on cutaneous pharmacokinetic studies, such as ones using dermal microdialysis or dermal open flow microperfusions that are in more advanced stages of development. And there are others that are in earlier stages of development, and we are still investigating them, such as cutaneous PK studies using imaging or spectroscopy-based tools, or the ones that is the focus of the today's presentation is to evaluate the impact of formulation differences on thermodynamic activity and sensorial perception of topical products to be able to expand the characterization-based approach to some of these test formulations that may have compositional differences compared to the reference standard. So we first needed to understand the function of excipients and their impact on thermodynamic activity of the drug in topical formulations. And we started with single-phase gels, which are the least complex form among the semi-solid topical dosage forms. And in the figure, you can see different types of excipients that may exist in a single-phase gel. We have solubility modifiers such as propylene glycol, polyethylene glycol, or glycerin. We have gelling agent, preservative, pH modifier, solvent, etc. And among these different types of excipients, we identified that probably the Q2 changes in solubility modifiers may have the highest impact on thermodynamic activity. So we started focusing on the solubility modifiers in our research. And in order to predict thermodynamic activity changes, we first needed to define fractional solubility, or alpha, which can be calculated from concentration of solute divided by saturation solubility of the drug or solute in the solvent. For example, if we have PEG water formulations, by increasing the amount of PEG in the formulation, since it is a solubility modifier, the solubility of the drug of interest most likely would increase. For example, here in this example, the saturation solubility of this drug has increased by increasing PEG concentration. But if we have a constant drug concentration, according to this equation, by increasing the denominator, the fractional solubility would decrease. So by increasing PEG concentration, the saturation solubility increases, but at 7.5 milligram per ml concentration, we have the lowest alpha with the 40% PEG formulation. And fractional solubility is often predictive of thermodynamic activity of the drug in the formulation and how likely the drug molecules would leave the formulation and partition into the skin. So one of the earlier studies that our collaborators did was to make series of propylene glycol water formulations. These were like 2-ingredient formulations, and the saturation solubility of diclofenac sodium, a lipophilic drug, was measured in series of this formulation with variant amount of PG from 0% to 100%, and as expected, by increasing in PG amount, saturation solubility increased. Then several of these formulations were selected in the range of 10% PG to 50% PG, and they conducted an infinite dose IVPT study. So as also explained in the previous slide, when we have a constant drug concentration, for example, in this case 2% diclofenac sodium, by increasing the amount of solubility modifier, we expected to see a decrease in the thermodynamic activity, and the IVPT results also show that with 50% PG, we got the lowest flux, steady state flux, and with 10% PG, we had the highest steady state flux. I would like to know that since we had infinite dose in this IVPT studies, the metamorphosis of the dosage form was negligible, but when the topical product is actually applied on the skin by human or by patients, the finite dose is used, which undergo metamorphosis and drying, and following solvent evaporation, there may be changes in drug solubility at the application site, or even change in microstructure and Q3 properties, for example, change in rheological properties of the formulation as the formulation dries, and all of them may lead to change in drug permeation and bioavailability. So while we can measure fractional solubility at time 0, we didn't know how this fractional solubility or thermodynamic activity would change across the product application on the skin. So what we did that our collaborators, they monitored the drug solubility or saturation solubility at the application site by measuring drug concentration in a donor compartment. So they basically had the Franz diffusion cell set up similar to how the IVPT is set up, and they used a semi-finite dose on the skin in the donor compartment, but instead of sampling the receptor compartment, they sampled the donor compartment and measured drug concentration in the donor compartment at different time points. So using this methodology, we would be able to have fractional solubility profile across the duration of application here, for example, for 6 hours. And here shows the results we got with HEC gel formulations, which included PEG 200 as a solubility modifier at different concentration, 1%, 10%, and 20%. And here the drug model was diclofenac sodium and with a fixed concentration. So as is shown here, with different amount of PEG, we had different fractional solubility, and during the course of 6-hour application, the fractional solubility with 1% PEG increased, and for the other two it kept constant. But what is important is we saw a well-separated profiles from the 1% and 10% and 20%, which suggests that the thermodynamic activity of the drug in these 3 formulations are different, and subsequently the IVPT results also showed that the permeation rate was significantly different from for the diclofenac from the 1% PEG formulation compared to 10% and also 20%. The similar experiments were conducted with HEC gels that contained metronidazole. So this is a hydrophilic molecule with different pKa and different physicochemical properties. And here, interestingly, with the variation in amount of PEG from like 1% to 10%, 20%, did not result in significant differences in fractional solubility at time 0 and also during the course of 6 hours. So subsequently, the permeation profile was also very similar for these for metronidazole from these 3 formulations. So we were able to establish a nice correlation between the drug bioavailability and the thermodynamic activity. Next, we wanted to focus on smaller Q2 changes and their impacts on bioavailability. So here shows examples of gel formulations where PG, propylene glycol, was used as a solubility modifier for both metronidazole formulations and diclofenac sodium formulations, and we first evaluated the saturation solubility of the drug in different amount of PG, and we for identify the reference product, we selected portion of this curve where it is relatively steep and it's within the range of PG amount used in approved topical products. So for metronidazole, the reference formulation contained 30% PG, but for diclofenac, the reference formulation contained 40% PG. And then from there, we calculated the PG amount in plus minus 10% difference and plus minus 25% difference in PG, and made those formulations with metronidazole and diclofenac. So the fractional solubility measurements showed that with plus minus 10% variation in PG, the thermodynamic activity is not changing, and subsequently the permeation rate is not changing compared to the reference formulation. But with plus minus 25% difference, the fractional solubility has changed, and the flux profiles also are distinguished from the one for the reference formulation. With diclofenac sodium formulation, while we see some separation of for the plus minus 10% formulation than compared to the reference, the IVPT result did not show significant difference in bioavailability of diclofenac for the plus minus 10% variant formulations and the reference formulation, but the profiles for plus minus 25% variant formulation were different compared to the reference formulation. Next, we wanted to understand whether the Q2 changes in topical products would have other impacts beyond changing the bioavailability. For example, would differences in Q1, Q2, Q3 of topical products result in differences on how the product would perceived by human subject and potentially change in therapeutic equivalence. So for that, we needed to establish a correlation between Q2, Q3, and sensory perception of topical products. And also we needed to understand whether the characterization of Q3 properties, such as rheological behavior of topical products, predict the sensorial differences that may be perceived by subjects. So for that, our collaborators did develop objective tools to measure some relevant Q3 attributes, and also conducted in vivo sensory panel tests of those formulations to be able to correlate these Q3 with those sensory attributes measured. So here we also started with single-phase gels, and we identified relevant sensory attributes of those gels. In the left image or figure shows the list of sensory attributes that we evaluated for topical gels, like spreadability, smoothness, shine, cooling sensation, etc. And we identified which of them potentially could have more impact on therapeutic equivalent. So for this study design, after identifying the sensory attributes, we needed to find for each sensory attribute what would be the relevant Q3 attributes that we can measure in vitro, and how we can measure that using what kind of instrumental technique, and also potentially what formulation variables can more impact the changes in these Q3 attributes and the sensory attributes. For example, for something like a cooling sensation, potentially a drying rate study was assumed to be a relevant test to inform about the cooling potential of a formulation, and probably the change in amount of solvents or co-solvent may have an impact on changing these sensory attributes. And for something like spreadability, we assume that rheological behavior of the gels, like measurement of zero shear viscosity, yield stress, etc., with a rheometer, would inform the change in the spreadability, and potentially the amount of gelling agents would have the highest impact in changing these Q3 attributes and this sensory attribute. So here are the results for the Q3 test. Some of them are shown here, and also the in vivo panel study for these three attributes that are shown here. For several carbopol 980 gel formulations that were made with different composition of a gelling agent and alcohol. And in this figure, the data is normalized for each unit, for each attribute, in the range of 0% dark blue up to 100% yellow. Formulation 2 here was the reference formulation. And for example, in formulation 3 and 1, we had differences in the amount of carbapol. Formulation 3 had like double the amount, and formulation 1 had half of the amount used in formulation 2. And these changes in the gelling agent resulted in significant change in the zero shear viscosity for these two formulations. But only in formulation 3, we observed a significant difference in the perception of stickiness and spreadability compared to formulation 2. And in formulation 4 that we had alcohol as opposed to formulation 2, the slipperiness of this formulation was perceived significantly differently. The similar studies was done with hydroxyethyl cellulose gel with different compositions of PG and propylene glycol and alcohol. Here, formulation 3 was the reference formulation, and the variation in amount of alcohol in formulation 1 to 4 resulted in significant change in zero shear viscosity. And in formulation 2, we had significant difference in perception of slipperiness. In formulation 4, where both the amount of propylene glycol and ethanol was different compared to reference formulation, the stickiness of the product was also felt differently. In conclusion, these preliminary results for this gel suggest that when we have Q2 changes, we may or may not see significant differences in sensory attributes. But when we have significant differences in Q3 attributes, we likely would see some, or we may see, or it's more probable that we see differences in sensory perception of those products. So with that, I summarize that FDA is investigating alternative scientifically valid methods, including in vitro approaches to support assessment of BE for topical products that have compositional differences compared to reference standard. The current research data suggests that when there are differences in thermodynamic activity, such differences may result in bioavailability as shown as evaluated by IVPT. The Q3 properties assessed instrumentally in vitro may be valuable in understanding most of the sensorial differences for topical gels in vivo. And the current data suggests that large differences in Q3 attributes, such as for like neurological, tribological, or textural properties, would likely perceivable to human subject. And finally, research is underway to further evaluate the impact of Q2 differences on bioavailability and product perception of topical gel formulations. So here are my challenge questions. The first question is, which statement is not correct? Q2 differences would always result in changes in performance of topical products. B, it may be feasible to assess fractional solubility of the drug in conjunction with IVPT to assess impact of Q2 changes on performance of the product. C, significant Q2 differences may result in changes in performance of the topical product. And D, metamorphosis of the formulation following topical application may change the microstructure of the product. So the correct answer is A, because Q2 differences would not always result in changes in performance of the topical product. The second question is, which of the following Q3 attributes is more likely to correlate with spreadability and stickiness of topical gels was based on the data I presented? A, friction of coefficient, B, zero shear viscosity, C, drying rate, D, all of the above. And the correct answer is zero shear viscosity. So with that, I would like to acknowledge and thank my colleagues and supervisors at US FDA and our research collaborators. And thank you very much for your attention, and we'll be happy to answer to your questions during the panel session. Thank you very much.